What if you understood enzymes and active sites by the end of this video? We are going to dive into the microscopic world of your cells and unpack some of the most fascinating and hard-working molecules in your body, enzymes. These are not just buzzwords from a biology class. They are the literal catalysts for life itself, making countless essential reactions happen in the blink of an eye. Think of them as the tiny, efficient workers orchestrating everything behind the scenes. So, let us begin with the basics. What exactly are enzymes? At their core, enzymes are biological catalysts, which means they speed up chemical reactions without being consumed in the process. Imagine you need to get from point A to point B. Without a car, it is a long, slow walk. Enzymes are like giving you a super-fast sports car for that journey, getting you there much quicker, and then being ready to pick up the next passenger. Why do we even need this speed? Well, consider all the processes happening in your body right now, digesting food, building new cells, repairing tissues, thinking thoughts. If these reactions happened at their natural, slow pace, life as we know it simply could not exist. You would be waiting years for your lunch to digest. Enzymes are the reason your biology operates in real time. The key is something called activation energy. Every chemical reaction needs a little push to get started, like a car needing enough energy to get over a hill before it can roll down the other side. That hill is the activation energy. Without enzymes, this hill is often too high for reactions to proceed efficiently within the body's normal temperature and conditions. And here is the cool part. Enzymes do not change the starting point or the end point of a reaction. They do not magically create new energy or remove existing molecules. Instead, they provide an alternative pathway, a shortcut that significantly lowers that activation energy hill. So, that daunting climb becomes a gentle slope, making the reaction much easier and faster to complete. To understand this better, let us use an analogy. Imagine a bustling factory floor. This factory needs to assemble a specific product from raw materials. Without any specialized tools or organized workstations, workers might fumble around, taking a long time to put things together. This is a reaction without an enzyme, slow and inefficient. Now, let us introduce our enzyme. In our factory analogy, the enzyme is like a highly specialized workstation with a perfectly designed jig or mold. Each workstation is specifically designed for one particular task, taking specific raw materials and transforming them into a particular product. They are experts at one thing and one thing only. This leads us to the active site. Think of the active site as the specific, unique pocket or groove on the enzyme where the action happens. It is the precise area on our factory workstation where the raw materials, which we call substrates, will attach. This pocket is not just any shape. It is meticulously contoured to fit its specific substrate. The substrates are the molecules that the enzyme acts upon. In our factory, these are the raw materials. Just like a key fits into a specific lock, a substrate fits perfectly into the active site of its corresponding enzyme. This highly specific interaction is fundamental to how enzymes function and why they are so precise in their roles. Initially, scientists thought of this interaction as a simple lock and key model, a rigid enzyme active site, and a perfectly shaped substrate. However, as we move forward, we understand it is a bit more dynamic. Think of it less like a rigid lock and more like a glove fitting a hand. This brings us to the induced fit model. When a substrate binds to the active site, the enzyme is not completely rigid. Instead, the active site can undergo a slight conformational change, molding itself more snugly around the substrate. It is like a handshake where both hands adjust slightly to get a better grip. This snug fit is crucial for the enzyme to do its work effectively. Once the substrate is nestled into the active site, they form what is called an enzyme-substrate complex. This is the momentary union where the enzyme holds the substrate in just the right orientation. The enzyme then exerts forces on the substrate, bending bonds, adding or removing groups, or bringing reactive parts closer together. It is like the factory worker carefully manipulating the raw materials on the workbench. Then something interesting happens, the chemical transformation. The substrate is changed into a product. This product is the result of the reaction, what the enzyme has successfully created. In our factory, this is the finished item, neatly assembled and ready to go. The enzyme has done its job efficiently and quickly. The final piece of the puzzle is that once the product is formed, it is released from the active site. And here is the beauty of it. The enzyme itself remains unchanged. It is like our factory worker who, after completing one product, is immediately ready to accept new raw materials and start the process all over again. Enzymes are recyclable, performing their function countless times. This means a single enzyme molecule can facilitate thousands, sometimes even millions of reactions per second. Think about that incredible efficiency. It is not just speeding up one reaction. It is a continuous, rapid-fire process that sustains all cellular activities. Without this rapid turnover, our biological systems would grind to a halt. What is fascinating is the extreme specificity of enzymes. Most enzymes are highly specific, meaning each enzyme typically catalyzes only one type of reaction or acts on a very limited range of substrates. It is like having a specialized wrench that only fits one specific size of bolt, or a specific key for one lock. This ensures order and precision in cellular processes. This specificity is dictated by the unique three-dimensional shape of the active site. If the active site changes shape, even slightly, it might no longer be able to bind to its substrate effectively or catalyze the reaction. The delicate architecture of the protein is absolutely critical for its function. It is a beautifully designed molecular machine. But what happens if this perfect molecular machine gets disrupted? Enzymes are delicate proteins, and their activity is highly sensitive to their environment. Think of our factory worker. They perform best under specific conditions. If those conditions change too much, their efficiency, or even their ability to work, can be severely compromised. Two major factors that significantly influence enzyme activity are temperature and pH. Let us examine temperature first. Most enzymes have an optimal temperature at which they function best, usually around body temperature for human enzymes. If it is too cold, the enzyme's molecules move slower, reducing the chances of collision with the substrate. The reaction slows down, like a worker moving in slow motion. However, if the temperature gets too high, things get much worse. The excessive heat causes the enzyme's delicate 3D structure, especially the active site, to unravel and change shape. This irreversible process is called denaturation. Once an enzyme denatures, it loses its specific shape and can no longer bind to its substrate or catalyze the reaction. It is like our factory workers' specialized tool melting and becoming useless. Continuing on, pH is another critical factor. pH measures how acidic or basic a solution is. Just like temperature, each enzyme has an optimal pH range. For example, the enzyme pepsin in your stomach works best in a highly acidic environment, low pH, while trypsin in your small intestine prefers a more alkaline environment, high pH. If the pH deviates too far from the optimum, the charges on the amino acids that make up the enzyme can change. This disrupts the intricate ionic and hydrogen bonds that maintain the enzyme's specific 3D structure. The result? Denaturation, just like with extreme heat. The active site loses its proper shape, and the enzyme becomes inactive. It is like pouring the wrong chemicals on our factory worker's tool, ruining it. What about things that can block an enzyme's activity? These are called inhibitors. There are different types, but the simplest to understand are competitive inhibitors. These molecules are very similar in shape to the enzyme's natural substrate, so they compete for the active site. Think of it like someone trying to use the factory workbench, even though they do not have the right raw materials for the job. Then there are non-competitive inhibitors. These do not directly block the active site. Instead, they bind to another part of the enzyme, causing a change in its overall shape. This, in turn, alters the shape of the active site, making it impossible for the substrate to bind effectively. It is like someone tampering with the factory workbench, making it unusable without directly sitting on it. The existence of inhibitors is not necessarily a bad thing. In fact, many are crucial for regulating metabolic pathways in your body. Cells need to turn reactions on and off, or slow them down, depending on what is needed at any given moment. Inhibitors are like the dimmer switch for cellular processes, preventing wasteful overproduction of certain molecules. Adding to that, some enzymes also need helpers to do their job. These are called cofactors or coenzymes. Cofactors are typically inorganic ions like zinc or iron. Coenzymes are small organic molecules, often derived from vitamins, like B vitamins. They can either bind tightly to the active site or loosely associate with the enzyme, assisting in the catalytic process. They are like specialized accessories or tools for our factory worker. Think about digestion as a prime example. When you eat a complex carbohydrate like starch, your body needs to break it down into simpler sugars. Your salivary glands release an enzyme called amylase, which starts this breakdown in your mouth. Then, in your stomach, protease enzymes like pepsin begin to break down proteins. Each enzyme is specifically designed for its particular job in a specific environment. In the end, the coordinated action of thousands of different enzymes, each with its unique active site, is what allows your cells to perform their incredible array of functions. From replicating DNA to generating energy, from detoxification to immune responses, enzymes are at the heart of nearly every biological process. They are the unseen heroes of your cellular machinery. The main thing to remember is that enzymes are biological catalysts that dramatically speed up reactions by lowering activation energy. Their magic happens at the active site, a specifically shaped region where the substrate binds. This interaction is precise, often involving an induced fit, ensuring that the right reaction happens at the right time. And their activity is delicately balanced by environmental factors like temperature and pH, and regulated by inhibitors and helpers. So, the next time you hear about enzymes, you will know they are not just abstract concepts. They are the unsung, tireless workers in your body's amazing biological factory, making sure everything runs smoothly and efficiently. Understanding them gives you a deeper appreciation for the incredible complexity and elegance of life itself. Pretty cool, right?